Thanks for inviting me. A lot of what I'm going to talk about is probably covered in greater depth in other technical lectures. So I'm going to kind of focus a bit more on why the models are useful, what are the kind of features of Gaussian processes that make them attractive for certain types of application. There is code for each sort of section, so you can dig into the details a bit afterwards if you're interested and actually run the code and try things out. But I'm going to focus more on kind of why we're using these kind of models and how we need to sort of adapt the models for the application. And so I'm going to take 4 kind of exemplar application areas, and they're kind of starting off pretty basic. And then as we go through, they'll get a little bit more complex, have a little bit more features that are sort of non-standard as we go through. So hopefully a little bit gentle to start with and getting a little bit more advanced later on. So I'm going to start off talking about something called differential gene expression. Often I'm talking to a biological audience and trying to teach them about GPs. So I had to adapt things a bit because I'm talking to a kind of more of a computer sciencey audience and trying to explain the biology a little bit. So do ask if everything isn't clear. I've tried to introduce things to keep things a little bit self-contained. So one of the fundamental things people are interested in cell biology or biology in general is how genes are expressed. So genes are sort of encoded within your DNA, and then the DNA transcribes into RNA, and then the RNA comes out of the nucleus of the cell, and it's processed in a few different ways that we'll get into during the lecture, and eventually often forms a protein. So there's this kind of flow of information from the DNA to the RNA and the protein. And today I'm going to talk about measurements of both the RNA level and the protein level. The DNA is, in most cells is the same. It's kind of static. It's, unless you're talking about something like cancer, generally it's just got the same information. But the cells and the human development or animal development is very dynamic. And that's driven by this process of gene expression. And so often what we do in biology is we measure this gene expression as it changes through time, and we end up with some kind of time course data. And by following gene expression through time, we hope to learn what are the rules by which cells decide which genes to switch on and off over time in order to sort of do what they need to do in order to have biological functions. So here's an example we'll look at later where we have some fly embryos. And so they're going kind of through different stages of very early embryonic development on the left. And then one view of that dynamic process is to look at the expression of lots of different genes. So we got genes there on the sort of going down the y-axis and time along the x-axis here. So lots and lots of genes, and some of them are switching on and then going off, and some of them are starting off low and then going up. And that's kind of what we call gene expression time course data. So it's very sort of common way of analyzing a biological system. And it has lots of similar features to other time course or time series data that you might look at. But it also has some sort of characteristics that make it challenging. There's a huge dynamic range. So genes might be expressed in just 1 copy on average per cell, or they might be expressed in 10,000 copies. So there's a big, big dynamic range. Big change in signal to noise, and there's lots of kind of sources of noise in the data, so you have to take that into account. So one of the most basic things people do when they're doing bioinformatics on this type of data is called differential gene expression, or searching for genes that change. So the data's maybe very high dimensional, so you can measure 20,000 different genes or so with these types of experiments. Very big differences in signal to noise. So before you do any kind of modeling and stick it into a machine learning algorithm, you might want to just filter things to work out which of these looks like it has some sort of signal. And a very kind of old paper about that presented this idea where you just do a simple kind of likelihood ratio test or a kind of Bayes factor type thing, where you have a dynamic model and a constant model. And the dynamic model can be encoded by a Gaussian process, and then the constant model is just a constant model, just white noise. And then you just look at that likelihood ratio and use that as a score to decide whether genes are dynamic. So that's super simple, but very useful. And it kind of avoids the parametric assumptions that a lot of methods that people use have. So a lot of people might use different types of generalized linear model to solve that problem. But the Gaussian process is nice and flexible. But in fact, modern ways of measuring gene expression use RNA sequencing. Yeah?
Sorry, just go back one slide.
Yeah.
Can you just comment a bit more on why you chose that statistic?
It depends, actually. You don't have to choose this statistic, but basically it's the marginal likelihood. So you're marginalizing out the GP, which minimizes the number of parameters. And then the dynamic model basically has, if I'm using RBF covariance, the dynamic model would have a length scale and a variance and a noise variance, right? So it'd have those three parameters. On the right-hand side, the constant model would have the same number of parameters, except it wouldn't have a length scale. And actually, the constant model is just a dynamic model in the limit where the length scale goes to infinity, right? So basically, it's nested like within the constant model. So the dynamic model is like the constant model is a special case of the dynamic model. So it means that if I take the length scale to infinity, that thing is 0. And if there's evidence in the data that the length scale is less than infinity, then that's evidence in support of it being dynamic. A frequentist would then do a likelihood ratio test. A Bayesian might do something different, like use a BIC or something like that. That's kind of the idea. Okay, but in that picture, I was doing a log transformation of the data, which is actually counts data. So the way you measure gene expression these days is that you do RNA sequencing. So you fragment up the RNA, and then you just count the number of fragments that you had. So the data you get is actually positive integers. And so it turns out that using a Gaussian likelihood isn't really appropriate for that kind of data. Now, you might think, well, all right, it might not be appropriate, but I'll just do it anyway. But it turns out that actually you can get a lot of benefits by modeling the data properly. And then people have done some studies of different types of RNA sequences and shown that it's pretty well modeled often by a negative binomial distribution, which is basically like a Poisson distribution, but where the rate of the Poisson follows a gamma distribution. So if you have a Poisson distribution, it doesn't quite capture the biological noise in your data sufficiently, so you need a little bit more variance. A negative binomial is like a Poisson with a bit more variance, basically. So you put a gamma on the rate of the Poisson, you get a negative binomial. And it's quite a good model for lots of this kind of RNA-seq data. So you can take that simple dynamic model, and you can stick the Gaussian process into the mean of the negative binomial. And then you have an additional parameter, the dispersion, which basically models the width in excess of a Poisson, right? And the reason that's good is because sometimes the counts are really low. Often they're 0. And it means that when you get counts in your data close to 0, the properties of your likelihood is a better model of the data. Whereas if you fit it with a Gaussian, if you log transform it and fit it to Gaussian, you're going to assume, basically, you're going to assume that everything is the same variance of noise everywhere, which isn't going to work very well. And you can't represent zeros. So here's kind of an example of what can go wrong. So in the bottom here, we're using a Gaussian likelihood. And this is what's called a two-sample test. So I'm now testing between two different conditions. Before it was dynamic versus flat. Now it's between two different experimental conditions. Short time series, not many replicates. So with not many replicates, it's quite hard to estimate the noise. In this particular data set, so there's green and blue conditions, the green ones, by chance, happen to have fallen quite close to each other. Right? So the data looks like it has really small variance, but it's just a kind of a fluke of the experiment, right? Because we haven't got many replicates. If you run it again, you'd probably get more dispersion in those points. So the Gaussian likelihood has decided to fit that data with a really small Gaussian noise variance. And by doing so, it's decided that it needs to fit the GP quite close to the data. And it's ended up with this kind of weird, wiggly green fit. And that's kind of led to bad behavior in this test. And then the one above is using this negative binomial likelihood. And because when the counts are low, it's kind of got a reasonable idea from the limit that it has to be at least Poisson, it's not going to kind of make that error. Right? This noise is at minimum Poisson, and it's expecting it to be a little bit more than Poisson. It gives it a bit of extra information about the noise and doesn't assume that the noise is the same everywhere. So it kind of fits things a bit better. So if you do this, and then you can benchmark this kind of procedure on data and check that this is also doing well empirically, and it does a lot better than using a log likelihood. Okay, so that's time series. And then the other thing people are interested is in spatial gene expression. And so one of the first ways of identifying spatial gene expression was work from Oliver Stego's group called Spatial DE, where they were using a Gaussian process and modeling spatial gene expression rather than temporal gene expression. And that's a really nice package. But again, it was assuming this Gaussian likelihood. So the idea is you have a spatial covariance model. You can also model things like periodicity and other kind of kernels there. And then you're basically scoring genes over whether there's really spatial structure in the data. So again, we can take that model and we can put the negative binomial on top of it. And it has two effects. One effect is it avoids false positives where the counts are very low because of this nice feature that you kind of revert to a Poisson noise with very little information. And then the other nice feature is that you have better sensitivity, so you pick up a lot more genes with this approach. And at the bottom here, on the left is this very sparse data that has barely any structure, but you can kind of pick it out with the GP. And on the right is some imaging from the same kind of serial sort of view of the data, but independent data to validate that there's really some spatial structure there. So it's a really sort of simple kind of use of Gaussian processes, but quite powerful, and yeah. Oh, so sorry. The grid there, on each point in the grid, there's a measurement of gene expression. So it's spatial in terms of like a slide. So basically, what you do is you take something like an organ, you'd slice it up, and then you'd measure the expression of the genes spatially. Yeah. So very similar to the time series, but now we're doing it in space rather than time. So this like very much vanilla GPs, but they've been pretty useful in this kind of domain. Actually, Oliver Stegel's lab since then has lots of nicer applications of GPs in that have kind of more sophisticated features. So he's really driving that kind of use of GPs in his group. But it's quite crucial to have a good likelihood in this application. If you just go with the vanilla Gaussian likelihood, you might come unstuck when you deal with things that have very low kind of integer counts. Okay, and the code for that's in the GP counts package. It was using GPFlow for those spatial ones and with single cell time series. You can have a lot of locations or a lot of times, so you need to scale things up a bit. So we use these sparse variational approaches. And actually, where's T gone? T was the one who helped implement the negative binomial likelihood in this model, which works very nice. And we've also implemented a branching kernel that I'm going to talk about later in this GP counts package. Okay, I'm going to move on to different things. So any more questions about that? That's kind of application of GPs 101, really, if you like. Quite straightforward. Yeah?
Is there some scientific reason behind choosing the negative binomial, or is it just like pick a big?
Yeah, there is actually. There's a bunch of papers which do sort of more first principles modeling of biological systems and show that there's pretty good reasons for it to be close to negative binomial. Actually, the history was initially with single-cell RNA sequencing, people thought that the data was very zero-inflated, right? So basically, they thought there were a lot more zeros in it than you'd see with a Poisson distribution. But it turned out that with more modern ways of sequencing, you can actually barcode the sequence reads so that you know which RNA molecule each one comes from with a little barcode. Once you do that, which is called UMI normalization, once you do that, basically it gets rid of the problem that the problem before was that you amplifying up the reads. So if something was 0, it didn't amplify up. And if it wasn't 0, it amplified up a lot. So you'd have something which was like a 0 and a 1, and it became like a 0 and a 10. And that leads to something being very zero-inflated because, or very non-Poisson. Once they did this UMI thing, they got rid of that problem, and then more modern protocols, the data has a nicer kind of, nicer structure, so it's more like negative binomial, basically. So there's quite a few, there are quite a few papers about it. So it's been shown both empirically and from a bit of first principles type modeling. Okay, so that was RNA. Now we'll talk a little bit about proteins. Okay. So another type of experiment that people do is they take proteins and then they heat them up. And when they heat them up, they unfold or denature. So they go from a folded state and they kind of open up into different configuration. And then they might form aggregates or do other things, right? So if you're cooking, if you're frying an egg, you have an egg, you stick it in the pan, you cook it, and the white goes white, and it changes. And it's not that it's got different proteins in it, it's just the proteins are forming complexes and aggregating and doing all sorts of things which they weren't when it was cool. So people do these kind of experiments because they're interested in discovering what proteins are interacting with. So they'll take a mixture that contains proteins. They'll add, say, a drug, like a small molecule, and they're interested in which proteins that small molecule is binding with. So they'll do this denaturization experiment and look to see whether this curve changes. Okay, so you got a control condition and a treatment condition. You do this heat treatment, you do your proteomics to quantify the proteins, and you do this in high throughput for lots of proteins at the same time for one treatment. And then, you know, the temperature changes, and the idea is that if the protein is bound to something, then it's going to change the temperature at which it denatures. So it's going to kind of give information about whether the protein is interacting with something. So the condition might be adding a drug to the mixture, and then if a protein interacts with the drug, it changes the point where the curve goes down. And, you know, so you want to measure kind of the change in this temperature where this denaturing happens. But some of the melting curves look very non-sigmoidal. So the sort of ideal situation is that the protein is denaturing and forming a kind of sigmoidal curve. But it turns out that you get very non-sigmoidal behaviors, and it's not noise because these things here are replicated. So each of these experiments has 2 replicates, and the replicates are behaving really nicely, right? So the experiment is replicating, completely different experiment, but you're getting this really weird melting behavior. And, you know, it's a little bit of a mystery why this is happening, but for instance, one thing that could happen is that the protein might be interacting with RNAs or DNA in the cell, so it's forming a complex. And then as the temperature changes, that complex changes as well as the protein itself denaturing. So it might be in a complex with something else, and it sort of disassociates from that and then unfolds or does something complex. So there's something weird in the data, and therefore, we don't want to necessarily make this assumption of this sigmoidal behavior. So we decided to really reduced our assumptions about the shape of the curve. So the sort of state-of-the-art method or the best package for analyzing this kind of data is called MPark, and uses fits a sigmoidal parametric function to the data basically and uses that to do the statistics. We decided just to use a Gaussian process, but we actually have lots of different information that we want to put into it, because often what happens is we're measuring lots of related peptides of the same protein, or we have lots of different conditions, like we might be trying out lots of different drugs, and we also have lots of experimental replicates. So we want to go away from the kind of parametric form, but we do want to share information across all the experimental data we have. So we're using a hierarchical Gaussian process, which kind of tries to capture the shared behavior of all the different conditions that we have and the replicates, while also being very flexible in terms of the shape. And I think you're going to hear about these tomorrow, probably. Is that right, Mauricio? Or is it going to be a bit of multitask type stuff? So I think you're going to hear about the details of this tomorrow. So this is an advert for this type of modeling that you can hear about tomorrow. And this is an application of it. So the idea is that you have a Gaussian process, but you have kind of different levels in the Gaussian process that capture different sources of variation in the problem. So you have your control versus your treatment, and then you have all these replicates. The replicates are correlated, so they're not kind of just treated as white noise, but they're treated as actually kind of correlated noise, because you get kind of these correlated, I want to say time, it's not time, it's temperature. Things that are correlated in temperature. And this is kind of implemented as a multitask Gaussian process. So just to write that down mathematically, you've got your kind of protein level, that comes from a GP, and then each condition is another Gaussian process centered at the higher level in the hierarchy. So each condition is like the general protein shape, but can differ because there's some condition differences. Then each of the replicates is like each of the conditions, but you might get some differences just because of experimental variability. And then you have some white noise, which is a sort of observation noise. And the whole thing is just a Gaussian process because it's just a linear combination of Gaussian processes. So everything is exactly a Gaussian process and can be treated in the same way. And similar to the likelihood ratio statistics I was using in the differential expression, we can look for differences between our hypothesis that the conditions are the same or the conditions are different. So, I mean, it was quite hard to empirically validate this because, well, okay, there's a semi-parametric Bayesian approach which uses a Gaussian process on top of a sigmoid. And there's a pure sigmoid approach in green, but using kind of Bayesian fitting. And those were clearly not doing as well. But our performance compared to MParc was quite similar, but the benchmark isn't perfect because we're looking for these things, this dark meltome, where people don't really know it exists. So we're kind of pushing into the unknown in terms of the shapes of these things. And we feel like the benchmark, so you're detecting true positives and false positives, we think it's probably conservative, right? So when we do these curves, we get the same results as MParc, basically, very similar. But it turns out that when we look at some of the examples that MParc calls as negative, they're clearly different. So in this panel D here, these curves, so we're looking for curves that look different. So some of these, MParc is calling them as this, basically it's failing on the test, so it's saying that those aren't different from each other. And the reason is that they're very far from sigmoidal behavior. So they're using the statistical test, which is assuming a sigmoidal, but if you show it things that aren't sigmoidal, it just can do something crazy sometimes, and the test statistic just doesn't work, you know, because it's very kind of strict model. But they're clearly different, and the GP fits are in the bottom right here, and the GP is really picking up very nice, very replicable across the replicates, very sort of quite noise-free type fits here. They're clearly different. You know, they're very weird, but they're clearly different. So it's really picking up genuine positives, we think, but they're not actually in the benchmark we're using for our ROC curves. So it's a little bit difficult to put a number on how many of these things there are because they don't all look as clear-cut as this, but we're definitely picking up things that are different. And when we look at them, they're often in the nucleus of the cell. And so we think that they might be sort of associated with these little speckles in the cell, these nuclear speckles, you know. So they're probably forming a complex or something because those speckles have lots of associations with other molecules. So we think there's some kind of signal in the data that we're picking up. So by sort of being a little bit less constrained in our thinking about what these denaturization curves would look like, we think we picked up some signal that you miss with the standard package. And again, there's code. This time we used GPyTorch. We used a kind of multitask Gaussian process regression. And the whole pipeline was kind of put together with Nextflow because we need to run it on HPC, and it's got other steps in it and everything. Yeah. Cecile, who put that together, has done lots of hard work on that because we want it to be used by sort of bench biologists doing that technology. Okay, am I doing for time? Okay. All right, over to the next bit. Any questions about that protein melting curve before I move on? Yeah.
How long does a project like that take to do from start to finish?
Yeah, I mean, that's almost her whole PhD, right? So that was quite a big project because so actually the complexity in that, so I was talking to someone earlier about this. So I'm started off very purely Bayesian. And so Cecile, who's doing that work, comes from more of like a frequentist background, the PhD student. And in biology, people are really often interested in false discovery rates and kind of controlling type 1 errors, the statisticians call it, right? So they're kind of interested in limiting the number of false discoveries in a long list. So they want to give the biologist 500 things, and they want to say, I think that 450 of them are correct, right? And 50 are probably wrong. And to control that in that particular data turned out to be really difficult. And in the end, we found out that the experimental procedure itself has lots of challenges that meant all the benchmark data sets that we were looking at were so the distribution of false discoveries didn't look right, basically. So there's a lot of work. So I think that her thesis will contain a lot of this stuff that probably the people who are behind that technology don't actually want to be in the publication necessarily. It's like a really sort of thorny. So she ended up working a lot with the actual experimental groups. Also trying to convince them to use a new package was quite difficult. So yeah, it's a real practical application. So that was the actual modeling. She probably got it going in a few months, but actually validating it and making it practical, that took a lot longer. Yeah. Okay. Yeah.
So you mentioned about the hierarchical model. So how long did it take to run the hierarchical model?
Oh, it's not too bad. This isn't like the computation time is like, a couple of hours or something. It's not like a massive application, but these experiments are quite heavy duty to do. So I think you'd be prepared to wait that long for the results. It's not like something you have to run interactively over and over. You just run it once to get the results. So I think the computation time for this one's okay. But for some of the ones I'm going to talk about later, that's a real problem, the computation time. So I'll get into that right at the end. Yeah. That's definitely a big issue in some applications. But this one's okay. Okay. So I'm going to go back to RNA now. I'm going to talk a bit more about time series, but with a bit more richer kind of models. So the data I showed earlier was from Drosophila embryos. And there's this really cool thing in embryos where the embryo initially is running entirely from the maternal gene expression. So basically, when you have an embryo, initially it's not producing any of its own RNA. It's just using RNA from the mother. And then there's a transition where the kind of embryo wakes up and starts producing its own RNA. But for a long time, it's just producing RNA from the mother. And we're kind of interested in studying that transition, which is called maternal to zygotic transition, right? Initially, you get all your RNA from the mother, and that's enough to make your embryo for a while. And then eventually, you have to start producing your own RNA. And then the other thing about that is that when the RNA is new, sort of newly produced, it has introns, and the introns have to be removed by a process called splicing. So you have this gene expression data here, And then you can see that for some genes, they're very highly expressed at the beginning. So those are maternal, and they're kind of switching off over time. And then some are low expressed early on, and then they go high expressed, and those are what we call zygotic, and they're kind of switching on. And here I've got 8 genes along the top here, and this one at the top left is maternal. It's sort of go high and then go low. Some of them go up and then down, so early zygotic, late zygotic. So you get all these kind of different patterns. And we're going to study the kind of zygotic ones. And basically, these intronic reads, they're going to kind of measure our pre-mRNA, this early RNA, and then they're going to get spliced out, and then we're just going to have this mature mRNA. So the RNA has 2 stages. You have your pre-mRNA and you have your mature RNA. The data on the left is measuring the pre-mRNA, and the data on the right is measuring this mature RNA. So the ones on the left are from the introns, so they're spliced out. And then the ones on the right, they're the mature RNAs that come from the exons. And you can see that the intronic ones precede the zygotic ones. But the pattern can look different from gene to gene because genes can have very different half-lives. And we're interested in what regulates that half-life. So we're kind of interested in what regulates the production of genes in general. And most people focus on what regulates the production rate, like how fast is the gene produced. But another way of regulating a gene is how quickly it's degraded. And if you think of a dynamical system, in order for something to be very dynamic, it has to have a high half-life. I mean, like if something doesn't have a high half-life, it means that you're just going to integrate the signal forever. It's just going to build up and up and up, right? So if you want something to go up and down a lot, if you want it to be really dynamic, it has to actually have a reasonably high half-life, because the half-life kind of sets the time scale for the dynamics, right? If you don't have otherwise everything just is going to build up forever, and it's not going to have, you're not going to be able to have good dynamics. So the half-life is quite important. So we model this by, we have production of these introns that are spliced out. So S is this rate at which they're spliced out. And then we have the production of the exons, the other part of the RNA, and they're degraded away by mRNA degradation. The 2 things are produced at the same rate because the way the RNA is transcribed from the DNA is the same for the introns and the exons, right? It's the same machinery is producing the introns and the exons. So we have these 2 linear differential equations, but splicing is quite fast. So we do something, which physicists might call adiabatic elimination. So we have 2 different time scales. The half-life of the splicing is fast, so we can kind of simplify things. And in the end, the production of the mRNA is proportional to splicing times the pre-mRNA. And then there's a degradation term at the end here. So it's a linear differential equation. Mauricio, are they going to learn about latent force models in any talks? Okay. So this is the simplest possible example of a latent force model, which is a way that a GP can be used to model a linear dynamical system, okay? Which kind of invented by Mauricio, actually. So. And those are a little bit more general than this. This is kind of how those things started, with simple differential equations. So the idea is that you have a differential equation, and we're going to model this pre-mRNA as a Gaussian process with, RBF covariance, so just quite a general, smooth function. Then the mRNA is a differential equation related to that pre-mRNA that we model with an RBF. And because the whole thing is linear, the solution to this differential equation is just an integral over p. And p is a Gaussian process. An integral is a linear operation, so it means that m and p are jointly correlated Gaussian processes. And that's kind of the idea of these latent force models, that you end up, this linear system ends up being a kind of correlated or multitask or multi-output Gaussian process. And here's a picture of a sample from this. So the blue here is this pre-mRNA, so that's the P. And the red is the M in this differential equation. And so the P is the production, and the red here is kind of integrating that because it's got a long enough half-life that it kind of integrates the blue signal. Okay, so here's two examples. So on the left, we have something with a long half-life. So here we have this red driving, so I've changed the notation, but that F is the P from before. So this is the sort of production rate, this red line. And then this black one is integrating that signal. And the peak of the mature RNA is slightly delayed relative to the peak of the pre-mRNA that's producing it, or that's nascent, the kind of new RNA. Then on the right-hand side, I've ramped up the degradation rate. And that means that the black line has come down because the degradation is higher. But you can see that the dynamics is more similar to the red one. So the peak of the black is closer to the peak of the red in this second one. Yeah? So basically, when you have a high degradation, the two functions end up looking very similar to each other. And as d goes to infinity, the black and the red would follow exactly the same shape. So the sort of degradation rate sets the delay between the peak of your pre-mRNA and your mature RNA. So if we take some synthetic data and we fit it to this type of Gaussian process model, we can then infer the hyperparameters. And for this, we actually used MCMC, so we used full Bayesian inference of the hyperparameters, because we wanted to get these degradation parameters with uncertainty in this problem. It's quite important to do things precisely in this application. And so we did Gaussian process inference as usual on the functions, and then we did MCMC on the degradation and the other hyperparameters of the model. This is synthetic data, so we know the answer, and we can see that we get good estimates of the degradation parameter that we're interested in here. So at the bottom here, the degradation rate is quite high, and the two functions kind of have peaks similar to each other. In the top one, the degradation is low, and the half-life is long, and you can see that the red one is kind of integrating the blue one in that case. So we applied that to this real Drosophila embryo data, and we found that there's quite a big dynamic range in the half-life going from, 3 hours kind of half-lives to a few minutes. So there's really a big dynamic range. And you can kind of also see that it makes sense. The ones which have a short half-life tend to be things that you'd expect to have to be quite dynamic. So transcription factors are these proteins that actually drive the gene regulatory networks of the cell. So in order for them to really do something on a short time scale, they'd have to have a short half-life. And then longer half-life, you tend to find things like receptor binding proteins that kind of bind to the surface of certain cell types in order to determine the nature of how that cell type defines itself. And those things you might expect to be more stable because they just have to kind of define what that cell type is. So I think it kind of makes sense, the categories that we're finding. And how important is it? So if we look at the dynamics of the pre-mRNA, the sort of nascent signal of the genes, we can see there's quite a lot of difference in the dynamics. So we actually used a Gaussian process clustering algorithm for this also from older work. And then if we look at one of those clusters with 210 genes in it, we can see that the degradation rate of the RNAs in that cluster leads to completely different shapes of gene expression within that cluster, right? So that's just one of the clusters which had a characteristic shape. Because of the half-life of the mRNAs within that set of genes, they themselves have very different dynamics. So it looks like the half-life is really determining the shape of the kind of gene expression response. So if you want to understand how gene expression is regulated in embryogenesis, it's going to be important to take degradation into account. If you don't, your kind of models for how gene regulation happens are not going to be capturing things correctly. Okay, and we kind of use that model to make some mechanistic discoveries about how mRNA degradation is regulated, but that's more of a biology story, so I'm not going to go into that. And again, the code's there. We use GPFlow again for this. And we use TensorFlow probability for the MCMC over hyperparameters. Just final thing is to talk a little bit about single cell data and branching dynamics. Often in biological systems, we're interested in systems where gene expression sort of changes between two different sets of cells or two lineages or two conditions. We can model that as a kind of branching process. And I'm going to start talking about time series and then talk a little bit about single cell. So what I'm calling a branching Gaussian process is one where you have a Gaussian process here in red, and then at some time, there's another Gaussian process that touches the red one but goes off by itself. So you have a kind of red Gaussian process, and then it splits into blue and a red one. That's one sample. There's another sample. And they can be very different shapes, but they kind of, have this branching nature. And you can model Gaussian process by considering like two Gaussian processes that cross a particular point, which I'm going to call TP. So if you have two Gaussian processes, F and G, and they cross at a particular point, then the covariance function is going to look like this, right? The bottom left and the top left and the bottom right are just your standard RBF covariance. And then on the off diagonals, you get this funny little kind of covariance which just says that two things are identical at a particular time, and then they forget about each other as they go away. So it's a covariance that just tells you that two things are forgetting about each other. So this is a covariance function for two curves that touch at a specific time. And then you can change it to be the covariance of two data sets that follow the same function up to that time. And then after that time, they forget about each other. They sort of go their own way. So you can sort of see that here. In the off diagonals, you've got the data following a normal RBF. And then it's kind of forgetting about the other function. So the two functions. And then there's this funny little kink in the bottom right. And that's just because, if I go back, that's not a good example. The blue one doesn't have to be follow the differentiableness of the red one. You could actually make that a little bit more. You could actually make the gradients agree if you wanted to as well, but we didn't bother doing that. So you can make it so that the first derivative has to be the same at that point. You have to stop somewhere, though. Like, if you make all the derivatives the same, they'll just follow each other forever with an RBF. So you've got that covariance. And then the inference tasks are, where's the branching time? If you've got some data, I want to pin down the time where they branch. And then, is there evidence of branching is another thing you can use a Bayes factor for. So here's applications to some time series data. So this is real gene expression data. And at the top, it's a posterior distribution of where they're branching, where the branching point is. And you can see it's quite clear that there's branching definitely, before 8. But there's a little bit of uncertainty exactly where the branching is in this model. And then on the right-hand side, the highest probability of the branching is at the end of the time window. And basically, if the bar on the right-hand side is higher than the average of all the other bars, then the most likely thing is that there isn't any branching in this kind of model. So you basically fit it for all the times, and then you basically integrate over all the times to work out these Bayes factors and posterior probabilities. Now, single-cell data has a little bit more complexity to it than time series, but people use something called pseudo-time to kind of infer where cells are in a dynamic process. They're not appropriate for all types of biological single-cell systems, but sometimes you might be looking at something that really does have a dynamics that you can infer. So the single cells that you're measuring might be all from different stages in some developmental process. So it's kind of like inferring time from high-dimensional time series data. And people use that a lot in analysis of single-cell data. And another story that I'm not going to go into is that you can use GPLVMs, another type of latent variable GP, to model that. And we've published on that, and others have also published on that kind of approach. But I just want to point out that when you're fitting the branching models, it adds a new inference problem. So in the time series problem, if I go back, I've got the labels for which dots are black and which dots are red. So I can so the whole thing is just a proper GP. Everything is a GP. There's no approximate inference required here. Right? You can do everything exactly. But in single-cell data, the cells aren't labeled with the branch they're on. Right? You're just given a bunch of cells, and you have to infer which GP the cell actually was generated by. So you have a new inference problem. You have to fit your branching GP to the data, but you also have to infer which branch the cell came from. And that makes the inference much harder. So in these kind of branching single-cell models, you have these new discrete indicator variables which you have to use to infer whether the cell came from a yellow branch or a purple branch. And it has to be consistent across all your genes. So we've got thousands of genes, and the yellow and the magenta and the green have to be inferred correctly for all of them. But we also have this interesting thing that the branching can happen early or late for different genes. So we came up with a GP model that solves the kind of problem of trying to find early branching genes in single-cell data where you don't know which branch it comes from, and you have to do all that inference at the same time. So it's kind of horrendous from an inference perspective. And we use a kind of mean field variational Bayes. So I really like this model, but I don't think that it's the go-to model for the single-cell data analysis community because of the computational burden, right? So someone was asking about computation time before. Yeah. So this is a problem for which that is basically the big issue, right? So people in this community of single-cell data analysis, they're dealing with bigger and bigger data sets all the time. This thing on 1,000 genes with thousands of cells, it's already taking hours to run. So modern protocols give you many more cells, and it really doesn't scale up so well. So I really like this model. I think it's an elegant way of solving this problem, but I don't think it's like going to get traction with the community, because I think the single cell community are driven by things that scale, because the data is getting bigger and bigger all the time. So elegant, maybe. Useful, not so sure, you know. But if someone can think of a way to scale it better, then that'd be nice. And for that one, we use GPFlow and TensorFlow. And the variational inference, we have these discrete parameters, so we have to use something called mean field variational inference over the discrete parameters within the sparse inducing variational thing. So yeah, there might be other applications of that where the data scale isn't such an issue, because I kind of like these branching models with the discrete parameters. But I need the killer application. Okay, so just to sum up, with the differential expression, the thing is that you're moving away from a parametric assumption, and a lot of people just use negative binomial GLMs for that type of problem with time series data, and I think the GP is a good solution. For the protein melting, it was more about sharing statistical power across many related data sets, replicates, conditions, protein variants. You want to kind of use the fact that all the curves you're fitting are similar, so you have to put that in your model. Degradation was a really simple example of this thing called a latent force model, where you have a simple linear differential equation, and you can use that within a kind of GP framework to infer your differential equation parameters. And then branching is nice for GPs because you have to integrate out the point where these two GP functions cross, and you can do it analytically with a Gaussian process. So that's why that's nice. But scalability in those single cell branching models is a massive problem that we haven't really solved. And that's all the people. The orange ones are biological, and the blue ones are more computational. And Jenny's like a hybrid. So that's why I use the colors. Okay, thanks. Yeah.
So in your first, in the beginning of your presentation, you used negative binomial to solve the zero inflated Poisson.
Yeah.
Did you ever try like have you considered like another like probability function like maybe Poisson mixture? Or
Yeah, actually, the package has a zero inflated likelihood in it. So it actually has a delta function at 0 plus a negative binomial in the package. So you can add additional zero inflation on top of the negative binomial as well. So you can have another parameter. But those recent works on zero inflation suggest that in that type of data, either that zero inflation isn't justified, or fitting it is so difficult that it doesn't make any difference. So zero inflation plus negative binomial, it's there, you can use it, but there's not really in that single cell and spatial transcriptomics. It's with most of the modern protocols, I don't think it's that useful, that's all. But you can do it. The code, you can write any likelihood you want in it, actually. You just need to work out the two moments of the likelihood under a Gaussian. So the code is super general. You can put in other likelihoods. Yeah.
so basically it's not practical for your case.
It's not that it's not useful for that case. The other Gaussian processes packages for spatial, they often use a Poisson with a white noise in the GP. So they'll have a GP, and they'll add white noise to the GP, and they'll put it through a link function and then have a Poisson. But I did see a blog post by one of the Stan guys warning about that likelihood, because if you have a big dynamic range in your GP, it depends on the link function. If your the link function is doing a lot of the work in deciding on the heteroscedasticity of the noise in that model, so you'd probably have to do some model selection on the link function. Like your link function can be log, or your inverse link function, or it can be sine plus, which is like linear and then kind of curves around. So depending on those two, you'd get different noise. So in a way, your link function is then determining your noise, which, I mean, that's okay as long as you're aware of it, because then you have to do kind of model selection on your link function, I think. See what I mean? Okay.
I think maybe you have another question. There was another question there.
Yeah. Oh, yeah. So in the single cell case, so in the first case, let me go back. There were two. So in this one, it's just a time series. So in this, it's just what they do is they take a time series, and then they might perturb the system. So there might be an immune response. And then the idea is you want to know which genes have an early response to the immune challenge. So actually, in this particular example, they took a plant, they knocked out its immune response, and then they exposed it to a bacterial challenge. And they had one plant that had an intact immune system and one that had no immune system. And then they wanted to know when the effect would affect different genes. And the idea is that the ones that are affected early are involved early on in the immune response. So they want to detect the genes that are first involved in the immune response in the time series. And then in this single cell data, usually it's to do with cells differentiating into different cell types. So they might be like early stem cells, and then the stem cells are differentiating into different types. So they have to differentiate, and then they go down different lineages, basically. And the idea there is we think it might be useful to work out which genes are driving that process early. So the ones where the change is early, we think they're maybe the pioneer transcription factors driving that change. That's kind of the idea. Yeah.
I think it's better to finish now. So
Yeah.
this,